Somatic motor neurons are the nerve cells that transmit signals from the brain or the spinal cord that control the action of skeletal muscles. So when you look at this image, this right here is two motor neurons. The word somatic that I used at the beginning, that simply means of the body. So these are nerves that control the contraction of these skeletal muscles. So the number of skeletal muscle fibers a single motor neuron innervates, and therefore the size of the motor unit is going to vary. And it can range from very small motor units that have less than 5 skeletal muscle fibers to larger motor units that may have several thousand muscle fibers. The size of the motor unit is going to determine the degree of control of those muscles and the way that they move. So this is what we call an inverse relationship, because the size of the motor unit and the degree of control, when one goes up, the other goes down. For example, motor neurons that innervate your eye muscles are small because greater control is needed to move the eye. In contrast, a motor neuron that controls several thousand skeletal muscles is in the power generating muscles in our lower limbs, our legs. That requires more power but less control. So again, a motor unit is a single motor neuron and all of the muscle fibers it controls. Again, remember, a muscle fiber is a muscle cell. The neuromuscular junction is where the nerve cell meets the muscle cell. Now look at the name of that, neuromuscular junction. Junction is where things come together. There's the nerve, neuro, there's the muscle. Neuromuscular junction, where they meet. It's usually somewhere around the middle region of a muscle fiber. Now, there are 3 parts to the neuromuscular junction. So we're just enlarging this right here. Okay, this is the synaptic knob. It's where the muscle cell, where the nerve cell enlarges at that terminal end. The synaptic cleft is the space between the nerve cell, synaptic knob, and the part of the membrane right here on the muscle cell that we call the motor end plate, because it's where the end of the motor neuron synapses with the muscle cell. Let's look at those 3 structures with a little more detail, starting with the synaptic knob, which is the expanded tip of the axon of a motor neuron. So remember when we saw that motor neuron on the previous slide, and let's just back up right quick if I can get it to go back for me. Well, I'm not going to mess up our video by trying to mess with it. So anyway, so here is the axon. This is what transmits the nerve impulse from the cell body down to the synaptic knob so that it can cause an action to happen in the next cell. So the synaptic knob, again, the expanded tip of the axon. Now, this houses a lot of synaptic vesicles. Remember, vesicles are little containers made up of cell membrane. They're filled with molecules of a neurotransmitter called acetylcholine. There are also calcium pumps embedded in the plasma membrane or the cell membrane of the synaptic knob. Those calcium pumps are going to be important in helping to maintain the concentration gradient of the calcium. So there is more calcium outside the synaptic knob than inside the synaptic knob. And the third component of the synaptic knob are the calcium channels that are also embedded in the cell membrane. Opening of those channels is going to allow calcium to flow down that concentration gradient from where there's more calcium in the interstitial fluid into the synaptic knob where there's less calcium, and that is going to trigger those synaptic vesicles to release acetylcholine into the space, remember, that's your synaptic cleft. The second structure of the neuromuscular junction is the motor end plates. Motor end plate. That didn't come out right. So it's a region of the muscle cell membrane where that synaptic knob or the synaptic terminal is going to come into that synapse. Okay, so it's that part of the muscle cell membrane right here that synapses with that synaptic knob. Synapse is just where they come together. Okay, so as I mentioned, the motor end plate is named because of the end of the motor neuron has like that large saucer-like or plate-like appearance. So the motor end plate actually has lots of folds and indentations of the membrane that increases the surface area that's covered by that synaptic knob of the nerve. So the motor end plate also has a very, very high number of acetylcholine receptors. Now, those acetylcholine receptors are chemically gated ion channels. So when the chemical acetylcholine binds, it opens the channels, allowing sodium to come into the muscle cell and potassium to exit. So the acetylcholine, again, does not enter the muscle cell. It simply binds to the receptors and works like a key. So the receptors are the doors. The acetylcholine itself is the key that opens those doors. Now that allows the gates to be open and sodium floods into the cell with a little bit of potassium exiting the cell. Now, let's continue with this one. The synaptic cleft is that fluid-filled space between the synaptic knob and the motor end plate of the muscle cell. In that fluid, there's a lot of an enzyme called acetylcholine esterase. Now, look at this word, acetylcholine esterase. ACE tells you it's an enzyme. The substrate is acetylcholine. So it's the enzyme that breaks down acetylcholine. Well, when acetylcholine is no longer in that synapse, it can't bind to those receptors, the gates don't open, and the sodium stops going in. Now, I want you to remember when we were talking about membrane potential in a previous chapter and how we can alter membrane potential by moving sodium and potassium across the membrane. So, yep, guys, that's where we're headed. So a very important feature of skeletal muscle is the electrical charge across the cell membrane, the sarcolemma. So right inside the cell membrane, there's a lot of negative charges. In comparison to the interstitial fluid outside the cell, there's a lot more positive charges. So that difference, as you can see right here, so here's the sarcolemma, the cell membrane. Look at all these positive charges right here in the interstitial fluid right beside the cell membrane. Now look inside the cell membrane and you see all these negative charges right beside the cell membrane. That creates that concentration gradient, the part of the electrochemical gradient. So this electrical charge difference is what is responsible for the resting cell membrane potential. The way I think of it is resting. The cell is not doing anything, but if we alter the charges across the membrane, there is the potential for that cell to do work. Okay? So the resting membrane is established and maintained by leak channels and sodium-potassium pumps. The function of those sodium-potassium pumps is to maintain that concentration gradient with more sodium outside and more potassium inside. Now remember also, the resting membrane potential is all about the positives outside and the negatives inside. To end this video, let's take a quick look at what's going to happen with muscle contraction. So this is an overview and we are going to go into a lot of depth. So I'm giving you fair warning, okay? Let's take this step by step. So the first thing that happens at the neuromuscular junction, the muscle cell has to be excited. Now remember, we talked about this previously with depolarization when we were talking about this in the earlier chapters. So the release of that acetylcholine from the vesicles in the synaptic knob, the acetylcholine gets into the synapse where it will bind to acetylcholine receptors. Once it binds to those receptors, then that muscle cell is going to start depolarizing because the gates open and that extracellular sodium starts rushing in. So let's move to number 2. The sarcolemma, the T-tubules, and the sarcoplasmic reticulum are all responsible for exciting the cell, leading to contraction. So we call that excitation, contraction, coupling. Again, that simply means excitation is depolarize the cell. Contraction, well, that one's easy. So the acetylcholine binds to the receptors, triggers the propagation of an action potential. That simply means the cell membrane of the muscle cell is depolarizing as sodium rushes in. Now remember, we talked previously about those T-tubules. So if you look at this muscle cell membrane, the sarcolemma, and you come all the way here to the green, you can see it is really just where the membrane comes down into the cytoplasm of the cell. So it dips down and surrounds the muscle fibers, okay? So because it is made of cell membrane, sarcolemma, just like out here, this depolarizes also. So think of it like you hit a domino over here, it triggers another domino to fall, another domino to fall, another domino to fall, and more dominoes to fall, 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 fall. Okay? Now, that is excitation. So the sarcolemma depolarizes, the T-tubules that are part of the sarcolemma depolarize. That leads to this purple, the sarcoplasmic reticulum, releasing calcium. So as we mentioned in the previous video, the SR stores and releases calcium, okay? So that calcium gets released. You can see all the little yellowish looking bubbles down here. That's representing calcium. So the calcium is released from the SR. It's going to come down here to the sarcomeres, remember, from Z disc to Z disc. It's going to bind to troponin. Tropomyosin rolls off the binding site. Myosin hooks up with actin, and the pulling of these two together shortens the sarcomeres and the skeletal muscle contracts. Okay, now I know that was a lot, but that is just our overview. I'm going to go back over it in greater detail. I'm actually going to show you some videos too, in addition to this video, so that we can understand this. All right, we'll pick back up with this on the next one.